Guys, so in this video we are going to do 2.3. It is the different instruments of microscopy, all of the different microscopes. So we're going to talk about all the different ones. I am not going to have you stress about the details about how every single one of them works. You don't need to know all those details. I don't know all of those details about how every single one works. But we do need to know generalities, the basics of how they work. So let's go through the different types and basically what are the differences between them? How do they use, are they using light? Are they using electrons? What are they using? So let's talk about those basics. So the first one we're going to start off with is light microscopes. If we were in face-to-face lab, you get to use the light microscopes. If you have done AP1 or AP2 or Bio 1010 or Bio 1107, you have hopefully put your hands on a light microscope. We had mentioned before that the light microscope can do a bright field, and that's normally what you're going to see when you turn on that microscope is a bright background, and then the organism itself is darker. Or there's a dark field. So we're going to go into both of these in just a second. Oh, sorry, parts of the microscope. I hope y'all already know this. I have them listed here for you, and I know for our virtual class that you don't get to put your hands on a microscope, but I'm really hoping that you have this experience already. Where are you looking through? You're looking through the oculars, and the oculars are going to magnify everything 10 times. So we're going to look through those oculars. Don't touch them to your face because you don't want to get pink eye from somebody else who also touched the oculars. That's gross. So that's why you clean your microscope every single time before you touch it. Oculars, you're going to grab and move around your microscope by putting one hand on the arm, and then you stick the other hand under the base of the microscope and you move it around. You're going to put your slide on the stage right here, and you're going to put the slide under side clips that will keep your slide in place. You will move the stage up and down with your focus knobs. So you've got the coarse focus knob and the fine focus knob, and that lifts it up and down. So that's going to be your focus. Coarse is going to move it a whole bunch real fast, and then the fine focus is the little bits when you're trying to just get in that nice resolution. It's your fine focus. Coarse is the knob on the outside. Fine focus is the inner knob on the inside. You are going to move the slide left and right using your stage knobs. They are going to be right here, stage knobs. Your light source coming down, shining up from the bottom up, not coming down, shining up from the bottom up is your light source. Then it is going to pass through a condenser over here, and then it will shine up through the stage onto your slide with your specimen on it. The condenser is going to have a diaphragm. So that iris diaphragm, it's just like on a camera, like really old school James Bond, where it can, go really narrow, or it can go really broad, really wide. So sometimes when you're in the face-to-face lab and you just cannot get it focused exactly the way you want, sometimes if you mess with the condenser and the diaphragm, that helps. If you change the amount of light coming through, that can help. That can increase contrast, and you can finally see the object that you're looking for. So don't forget that condenser and the diaphragm is underneath there. So you're going to look through your object. You look through for the oculars. And then you have different objective lenses. That's these guys right here that are housed on, they're dangling down from the nose piece right here. So the different lenses, the lowest power lens is called scanning. So anytime you put a slide on deck, your stage should be down low. You're going to put your slide on there, and you're going to make sure that your objective is on scanning. And then you're going to crank with coarse focus it up to where you can see your object under scanning power, or at least some sort of blurry something there under scanning. You start on scanning first, and then you will move up through the objectives and get higher and higher and higher magnification. Scanning's magnification is only 4. You will move up from low to high. Low is 10 and high is 40. all the way up to, in this class, in micro, you get to use oil immersion, which is something that we didn't get to use in AP 1 or 2. Oil immersion gets you to 100 times magnification with that oil immersion objective. So if you combine that oil immersion objective with your ocular that you're looking at from the top, the ocular was 10 and the oil immersion objective is 100. So when you have to magnify it, you times them both together. That's your total magnification. So 10 times your 100 would be 1,000. So a light microscope can magnify something 1,000 times more than you would see with your naked eye. That's as high as it can go, is 1,000 times on a light microscope, 1,000 times. That's as high as it goes. So we can still see small things. We can make out bacteria. We can make out red blood cells and white blood cells. But that is our limit on a light microscope. So here's our oil immersion. Why do you use oil immersion? Well, the lenses, the objective we said was 100 times, but when we add the oil in, so you're going to move your objective out of the way, so there's nothing in the way. You're going to place 2 or 3 drops of oil onto your specimen slide, and then you push the objective from the rotating nose piece into place, and your oil objective is now going to connect with your drops of oil that you put on your object, and now we've got this oil kind of wall that is attracted to the objective lens with capillary action, and it's stuck together. So what that does when the oil droplets connect between your specimen and the objective lens is now, instead of the light going through the glass and then it gets refracted, remember we said it bends, that oil is going to capture the light beams and keep them going straight, so you get a better, clearer image with and higher magnification when you use the oil on the oil immersion objective. So this is your light microscope on the oil immersion objective. It's 100 times for the objective lens, and then you're going to multiply it times the 10 on your ocular for your total magnification of 100 times. If you were using scanning, you would have 4 times 10 and you'd have 40 total magnification. So this is light microscope, how it works, total magnification and parts. I'm hoping that all of this is a repeat for you and you already know all this stuff. Bright field is going to have, again, the white background with the dark object in the middle of it. Dark field is going to be the opposite. So the background is dark and your organism is light. And that's because you're going to put in something called a light stop down here before it hits that condenser, in between the light stop and the condenser lens, before it goes up to your sample that's up here on the slide. And so that is going to, you can see it, the light stop blocks the light waves in the middle, and it only gets the ones out on the edge, and then that's going to hit the condenser, which is going to condense it into a focal point, and that focal point hits the object. So what you see is the object that is either absorbing that light and then it refracts it back, and you can see the structures better on that organism. A nice thing about the dark field microscope is that it gives it an increase in high contrast, it's high resolution, and you don't have to stain the objects, because oftentimes when you stain something, you have to, heat fix it, you can put your stains and your dye on there, and that's going to kill your organism. So if you're interested in looking at your object that's still alive, then dark field microscopy might be an option for you. So here we have, what are we looking at? Oh, Trypanemia pallidum. So if you were trying to look at syphilis in a specimen, you might want to see these guys, and you can see they glow and the background, the field is dark. So this is dark field microscopy, and it's a great way to look at live specimens because you didn't cover it with stain that's going to kill the thing. Okay, other types of microscopes. Phase contrast is going to use refraction and interference. So remember when I was telling y'all about how you drop the pebble in, and if you skip the pebble across the lake, then each time it hit and skipped, it was going to cause its own ripples, and those ripples interact with each other and cause interference. That's how phase contrast is. So it's going to alter the wavelength of the light rays coming through. And when you alter that wavelength, the light waves coming through, you end up getting a better resolution for this. So it's an increased contrast without using stains, with this phase contrast microscopy. So it's pretty cool. I'm not going to ask you about the details on phase contrast. Where exactly is the annular ring placed? So don't need you to know those kind of details. But I do want you to know that phase contrast is done by altering the wavelengths, and it is a great way to look at a specimen without having to stain it, and it's going to increase your resolution. So here it is on a bright field, and it looks okay. Yeah, it's all right. I can see that there's something there. We got this round blob here, and then we got this thing here and there. But then if you look over here with the phase contrast, you got way more detail with the phase contrast microscope. So it's pretty cool to see this difference, and you didn't have to use an electron microscope. You didn't have to add any stains to it. This is phase contrast. Differential interference contrast. Interference, again, similar to phase contrast. It's going to use interference patterns. It is going to use two beams of light on a DIC microscope. And we can see this beautiful picture of this fungus that is growing here using our DIC microscope. So high contrast organism pictures of living things. Fluorescent microscopes. So we were talking about fluorescent dyes and how they can take energy from a higher level like UV, and then they can transform that energy. They'll absorb that energy, and then they will reflect out a lower level energy that you and I can see with our eyes. So with fluorescent microscopes, we got an energy light source. It's going to send in a higher level energy like UV or blue that's over there on the higher energy spectrum. It's going to send that in. It's going to hit the specimen, and then that is going to activate those fluorescent dyes that we put on the specimen, right? And then those fluorescent dyes are going to transmit that high energy into a lower level that we can see with our naked eye. And that's what we're going to see on the detector. So those are fluorescent microscopes. We can use fluorescent microscopes when we're talking about immunofluorescence, and that is going to be a test that we'll talk about later on in the semester. But it's where we're trying to see if antibodies are attaching onto an organism. And we want to know, is this sticking on this organism? Are we looking at the right organism? Because if it is, that antibody is going to attach to it. If it's not, the antibody won't attach to it. And we can use immunofluorescence to detect this. Confocal is going to be our next microscope. This one is going to use Z-planes. It's going to use these two-dimensional images of these Z-planes, and it's going to piece them together. It's kind of like X-rays and CAT scans. So X-ray is just one image, right? And we put them all together and we can get a 3D image. That's kind of how confocal uses these. So it's going to produce these 2D images at high resolution, and we put them all together, and the end result is a 3D image. Two photon microscope is going to use scanning technique, fluorochromes, and long wavelengths. So instead of our fluorescent microscopes that used high energy, high frequency wavelengths, the two photons are going to use lower ones like infrared. That's down on the lower end of the spectrum below red. Infrared is going to be a 2 photon microscope. And so these things are expensive microscopes that we don't have on campus. We don't have any of these other guys on campus. So these are really cool. They are going to be less damaging if you use this type of microscope because of that long wavelength, because we said the higher wavelengths, the higher energy, the more damaging those are to cells. So it's really cool to look at living things, intact tissues, and that kind of stuff. So that's 2 photon. And then we have the electron microscopes. Electrons can be transmission either way. If it's an EM, if you see EM at the end, they're using electrons. Electrons are going to be short wavelength, high energies. That is going to be, we said earlier that short wavelengths are going to increase resolution, and we're going to have a better magnification too. So electron microscopes, if you see a great image and you see TEM for transmission or you see SEM for scanning electron microscopes, they are using electrons and they are either bouncing it off the surface or they are transmitting it through the object. So if you were looking at external structures on something very small, then it's an SEM. If you were looking at the internal structures inside of that bacteria, then it's probably TEM because it's transmitting through the object. I don't need you to know the steps on exactly how it works, just generics that they are using electrons, know the difference between scanning and transmission. That number is off. Oh, okay. I'm like, wait a minute, that's not right. The light microscopes, we said total magnification was 10 times the 100 on the oil immersion to give us a total of 1,000 times what you would see with the naked eye. Electron microscopes, again, they're using electrons with a higher energy. They get increased magnification, increased resolution. So they are now at 100,000 times. So they are seeing much smaller things on an electron microscope than a light microscope. TEMs versus SEMs. And the imagery, right? Our scannings versus our transmissions. Are we looking at, look at all these internal parts that you're seeing here. Look at all these little guys that you can see these structures inside. That's going to be a TEM image if we can see what's going on inside because it transmitted through. If we're looking at the outside, at these lovely spherical shaped guys, these cocci, and we're looking at the external structures, maybe these things we need to know what protons are using, what proteins are using to attach onto stuff, then maybe we're looking at a scanning electron microscope image. All of the electron microscope imagery is black and white, and then somebody comes in later and colorizes it, makes it look pretty. Biofilms are something that we will talk about later on, but basically they are accumulations of different bacteria within like a, you know how I showed you the grand prismatic spring and all of those different colors? Those were mats, those were biofilms. And so it's just layers of bacteria on each other. How do you look at one of these? To look at these structures, confocal and fluorescence would be great microscopes to use to look at a biofilm. Here it is with a DAPI stained biofilm and what it looks like using this on stainless steel. And then our last guy is the scanning probe microscope. Before, what was that number again? It was 100,000, right? When we were talking about, yeah, the electron microscopes. Oh my goodness, electron microscopes are 100,000 times, while the scanning probe, and it's just like it sounds, it's going to use very sharp, teeny tiny little probes that are passing over the surface of something. So I am passing over the surface of my hand and I can detect the different shapes on it. This thing can go to 100,000, right? No, 100 million. 100 million times. So we are getting so much stronger. So our scanning probe isn't using light or electrons, but these very sharp probes that can interact with the specimen and crazy small. We are looking at very, very small things like atoms with a scanning probe. So here are the different types of things that we can look at all in one table. The OpenStax likes to put a whole bunch of information in these tables at the end, and I will refer to these too because they're kind of like the nice like cheat sheet, cliff note kind of things that gets all the information in one place. So we got all the different types of microscopes in these tables, and you could see, excuse me, some really good images from all these different types of microscopes here in these tables. Okay, so Cindy was the camp counselor with the scraped up knee and the infection that didn't seem to go away. So remember, the counselor sent it out. She wants to send it to somebody else and have them culture it, see what's going on. So she sent it to the nearest lab facility, and they're out at a camp in a remote rural area. So the lab that she sent it to was poorly equipped, and it did not culture and grow this stuff. In this place, they just took the sample from Cindy that was sent in from Cindy, and they just stuck it on there and made a wet mount and looked at it under a bright field microscope. So when you wet mount something, if you took 1107, I think we do this in 1010 as well, maybe. You take your glass slide, you add your specimen to it, and then you add water on it, and you pop on a cover slip, and you put it under your light microscope. Hopefully all of us have been able to do this. So that's what they did with Cindy's stuff, and looked it under there, under the light microscope to try and see what they could see. They could barely see the bacterial cells because it was clear they just did a wet mount against a bright background. So they are going to put in a light stop. Remember what happens when you put in a light stop? What does it become then? Now it is a dark field image, and they can see the bacteria better that way. So these bacteria are spherical, and they are grouped in clusters like grapes. So we told you earlier that you do need to memorize bacterial shapes. So shapes that were spherical were cocci, and if it is growing in a cluster, that tells us something. So we can group things if we know characteristics about different things, like Staphylococcus, Streptococcus. They're round-shaped organisms, and so that tells us a little bit about ourselves. So they are seeing spherical, cocci-shaped bacteria, and they're grouped in clusters. So that's another thing. How are they growing? Are they growing in a cluster? Are they growing in a chain? Are they individuals and they're in singles, or in a packet? How they grow is also another indicator of what you might be looking at. So these are spherical, growing in clusters, and that's what we know so far about the bacteria found in Cindy's knee wound from playing basketball. All right, I'm going to stop this video on microscopes, and then we'll talk about staining techniques in the next video.